So see if you can do this from memory. Don't look at your notes, right? So for the reproduction of a virus, what's the very first thing that needs to happen? Attachment, right? It must attach to the cell's surface, right? And we're going to look at that. We started looking at that last time. We're going to look at it again today. And then we're going to go into the details of what happens next. Entry, right? It's got to get into the cell. And clearly, if it can't do that, then if it can't do either one of these, it can't cause infection, right? If it can't attach and if it can't enter, right, you don't get a viral infection. But once it enters, what needs to happen? Replication, right? So I added what I didn't have there before, right? So they're going to replicate their viral proteins and their viral nucleic acids. How are they going to do this? They're going to use the machinery within our cells and the building blocks within our cells to make them, right? So your cells basically, or the cell that gets infected, basically becomes now a virus-making factory. Okay? And then, of course, right, the nucleic acids get packaged into the proteins, and then if you have an envelope, that needs to happen. But that doesn't actually happen at this phase, right? Self-assembly, they're talking about just the viral proteins, the protein coat surrounding the nucleic acid, right? Forming the nucleocapsid. It's actually this last step, so of course, once you've made the viruses inside the cells, they need to what? Get out of the cell. They need to exit, right? So you need release of those prodigy virons out of the cell. That's the phase in which the envelope gets put on. Because the envelope is actually a modified membrane from the cell it infected. And so hopefully we'll get to that today. We will see. Okay. Yeah, we'll see. We shall see. Okay. So, right, diagrammically, right, the steps. And unfortunately your book kind of jumps around with us between the chapters. So we talked about this last time, but just to review, right, the reason why certain viruses only infect certain cells is, again, that attachment phase, right? That protein on the virus has to recognize a protein on the surface of the cell it's going to infect, right? It's literally an attachment and interaction. And the problem is that the receptors or the proteins that virus is attaching to are usually critical ones. So we can't avoid infection by changing those, right? And we'd have to change that at a genetic level. And we talked about an example of that today last time that we'll talk about again, where at the genetic level, right, there's people in Africa who their receptor has changed enough on their cells that they're genetically immune to HIV. So we'll look at, I know that because that's of interest, right, and it sticks in y'all's heads. That's one of the ones we'll look at today, right? So this is the first receptor, right? That's where the HIV binds. So for a lot of viruses, right, we know what receptors they bind to. I don't expect you to memorize any of them, right? But again, for any type of virus, whether it's one that infects bacteria, bacterial phage, notice its tail fibers, right? Those proteins are interacting with receptors on the cell, right? If it's an envelope virus like the influenza virus that has protein spikes in the envelope, those protein spikes are interacting with a receptor on our cell surface. If it's a naked virus, right, but also has spikes off of its vertices, those are proteins, right, that are interacting yet again with receptors on the surface of our cells, right? Adenovirus is the example they give you here of that type of virus. And then some of them just have the protein coat and no spikes, but the protein coat is just that. It's protein, right? So it's proteins within that coat that are directly interacting, and the example they give you here is poliovirus. All right, these are non-enveloped viruses, right? But again, that's protein. So we talked about last time, if that receptor changes, right, then the virus can't bind and you're genetically immune. So they give you a mouse example. We talked about that HIV example last time. So okay, attachment, that's pretty straightforward, right? Proteins on the virus, right, on the outside of the virus, attaching to receptors on the host cell. Okay, now we have to get inside that cell. There's some barriers that some cells have that others don't, right? So plants have what additional barrier that a virus would have to get through? The cell wall, right? Every cell has a cell membrane, right, which the virus is going to have to get through. But for plants, right, they've got to get through that plant cell wall first and then into the membrane. Then past the membrane level. For our cells, right, vertebrate, most animals, right, do we have cell walls? No. All they have is the membrane to contend with, right? So all they have to do is get through the membrane. Bacteria, we know, have a similar problem, right? And that they have what? Like plants. They have a cell wall, right? So again, their viruses have to deal with how do you get through that cell wall. And plants, viruses, and bacterial viruses, this happens differently. So obviously probably the most important step, right? Because if they don't enter the cell, they can't replicate in the cell, okay? So animal viruses don't have to contend with that cell wall structure. Oh, I forgot about fungal cells. But also algal cells, right? Algae have cell walls. They're basically microscopic plants. All of these have to, all of them have a cell wall that the virus would have to contend with. So remember, I pointed this out last time in your book. This diagram, they have interactive where it kind of steps through the different ones. So I even put a note here where that is in our book, so we could find it and zoom in on these. So I'm going to go to that. And unfortunately, I can't have more than one section of the book open at a time. So I'm trying to remember, because I want to come back to this one, because again, we love to talk about HIV. I need to make a note of this one so I can find it later. So I might want to come back to 8.3 later, section 8.3. Okay. But I want to go to chapter 5, section 1 right now. Got to go to 5, Erica. It has to load. Section 1. Alright. And so it starts at the bottom. Here it is. Right? So here's the figure, but below it, here's our interactions where we can kind of step through. Let's see if I make this full screen comes out good. Okay. So, alright. So if you have, and let's look at this virus. What type of virus do you think that is, looking at that picture? This is a rhinovirus, causes common cold, right? So what do you think this squiggly line represents? The nucleic acid, right? I don't know if it's DNA or RNA, right? And then this is that lovely protein coat. And what do we call this very common shape that you could have made origami homework for? Isohedral, right? So this isohedral shape. So this protein coat, the capsid, right, is attaching directly to this receptor on the host cell. When that happens for this particular interaction, it actually causes the host cell to use its membrane to surround that virus into what's called an endosome. Right? So that interaction is And our cells do this, right? They'll take stuff in, like they'll surround their membrane around it. So this particular receptor interaction induces this type of response, right? Where it's going to use the membrane to surround it within an endosome. So this is endocytosis, right? We talked about this a while back, right? Except this is not the type of endocytosis we like, because we're taking in something bad, right? So then that virus is inside a membrane, right? Inside of our cell. Well, it can't hurt us if it stays there, right? So obviously it needs to get out, right? And that's the next step, is it's going to escape that membrane, and the nucleic acid and the protein coat must come apart from each other so that this information, right, this is the information, can be used to make more protein coats, right, and more nucleic acid. That's the synthesis phase, right? So part of entry is them coming apart. This is sometimes referred to as uncoating, right, because the protein coat, right, comes apart from the nucleic acid. All right, so now let's look at the next one. So that's one possibility. That's referred to as receptor endocytosis of a non-enveloped virus, right? So naked virus. The other thing that can happen and I choose HIV for this one, right, is an envelope virus, right? HIV is an envelope virus, and so it has protein spikes in that envelope, though, that interact with a particular receptor on our helper CD4 T cells, which are very important in our immune system, unfortunately. That induces binding with one of these co-receptors here, and in doing that, this membrane, right, so this virus comes down and touches, let me zoom in, right? I forget I can zoom in, right? So then folds over, attaches to the co-receptor. Right, together, this has to happen. And so the people that are immune genetically in Africa, it's this co-receptor that's different. It's actually not their CD4. Okay, and then the virus membrane touches our membrane, and the membranes fuse. And so then the nucleocapsid, right, so this is the nucleic acid surrounded by the protein coat, enters into the cell, and then the two of them separated uncoats. And synthesis begins next. So that one is referred to as, again, right, this one, because it fuses with the membrane, this is referred to as membrane fusion of an enveloped virus. And really only enveloped viruses can do that because they're the only ones that have that envelope that's very similar to a membrane, so the two can fuse. Okay? Who had a question? Oh, okay. Sometimes you just have to give me a minute. All right, so the next one that we're going to look at is endocytosis, but now the virus is what? It's endocytosis again, but the virus is what type of virus? It's the influenza virus. And look closely. This is an enveloped virus, right? So notice it has an envelope, but that envelope does not fuse with our membrane. Instead, the receptors it binds to induces endocytosis, just like what happens for naked viruses. So they get taken into an endosome, and this is when the fusion happens, inside the cell. Do you see that? The virus's envelope fuses with the membrane of the endosome, and this releases the nucleocapsids. Because remember, each one of these for influenza is a piece of RNA surrounded by protein. So they don't show the uncoding process here for this particular one. So these are the ways it can get into cells that have membranes, like our cells, right? And as I said in the beginning, of course, most of the viruses we actually came in contact were enveloped viruses. And then we discovered the ones that don't have the envelope. So in summary, right, if you have naked virus, non-enveloped virus, it can bind to the receptor and induce endocytosis. That can even happen with enveloped viruses, right? But to get out of the endosome, the envelope actually fuses with the membrane in the uncoding process. Or, right, for envelope viruses, the envelope itself can fuse with the cell's membrane, and the nucleocapsid, the protein, the nucleic acid can enter right in. So 2 possibilities for enveloped viruses, right? Just really only 1 possibility for non-enveloped viruses, just endocytosis. Okay, so for plant cells, there has to be some type of injury to that cell wall. And it actually doesn't take much to damage the cell wall of a plant, apparently. Rain, hail, right, wind, depending on the stability of the plant. The most common way, of course, the first on the list, is insects that feed on plants. They can transfer the viruses from plant to plant. And then once it's into a cell, cells are connected in plants, so then it spreads cell to cell, and doesn't have to worry about the cell wall anymore. It takes advantage of those connections between the cells that exist. One of the ones I find most interesting too is fire damage, because when I was in graduate school, one of my classmates, they weren't the molecular ones, they were the ecology ones. Their dissertation, there is a pine tree here. I forget the name of it, the type of pine trees here. That their pine cones don't actually release the seeds unless they're burned. And the trees themselves, the bark is very fire resistant. So it's actually, fire is necessary for their life cycle. And of course, lightning can start fires, right? So that's the natural order. But again, a lot of times we try to prevent fire from happening, of course. But for these trees, they have to do controlled, purposeful burns. Otherwise, their reproductive cycle doesn't happen, right? So it seems kind of strange, right? You know, that fire could be a good thing. But as far as viruses go, right, fire could damage the cells, and then of course, we damage plants as well, and animals. So for bacteria entry, right, we're dealing with that cell wall. And I didn't have time, but you can probably YouTube it. At one time, one of my students found this really cool animation where they show this animated. But it's literally looks like a spaceship, right, landing on the surface of the bacteria. And again, there's that attachment of the tail fibers. And then the tail comes down, right? So literally like landing. Has these spikes that poke through, right, the cell wall. And so therefore the nucleic acid is the only thing that enters into the cell. But that's all it really needs is the information, the DNA. And any enzymes they may need, right? But the whole virus itself doesn't enter in. It just injects the DNA through that cell wall. It's pretty cool. But then from that DNA, right, synthesis is what happens. They copy the DNA, they make these proteins inside. Okay, so I was trying to decide earlier if I wanted to do this one first or jump ahead and come back to this. So I'm still in debate. I think I'm going to jump past it for now, and you'll see why. We're going to come back to it. Okay, so we've already talked about this, right? Just a little bit of a review, right? The DNA can be double-stranded or single-stranded, right? Or it can have RNA. It can be single-stranded or double-stranded. It can be linear or circular. That's something we haven't talked about yet. The problem with if you have single-stranded RNA, which is, what's in our cells, it depends on directionality. If you remember back, right, from biology class, maybe, that enzymes that copy DNA and RNA move in a particular direction. So, RNA has a directionality to it. And so one direction is referred to as plus or positive single-stranded RNA. And the way I remember this and link it up as to why this one's the good one, I guess you could say, is that this is the same as messenger RNA in our cells. And so this is what the ribosome can bind to and make proteins from. So if you're a single-stranded RNA virus of the plus strand, that direction, the ribosomes can directly attach to your RNA and read it and make those viral proteins. Negative strand, it's the wrong direction, right? So ribosomes cannot directly attach to negative or minus strand single-stranded RNA. So that's a problem, right? Because the virus needs our cells to make its proteins. Well, if our ribosomes can't bind to their RNA, what additional step are they going to need to do, and what additional enzymes that they need to come with? They've got to turn that minus strand RNA at some point into plus strand RNA, right? So that it can be read by the ribosome. So that's when I show you a scary picture pretty soon. So, oh God, his name's not on here. Shame on me. This classification system is, of course, named after the guy who came up with it. So viruses aren't only classified by whether they're DNA or RNA, single-stranded or double-stranded. They're classified on how that information is copied and utilized by the cell, right? So in viruses so far, we found that there are 3 different types of DNA viruses. And there are 4 different types of RNA viruses. So guess what? He created a classification, 7 different groups. 3 plus 4, right? I know it's early in the morning, right? Y'all got that? Okay. So then when you see this, right? Baltimore, I don't know why I couldn't remember Baltimore. I could remember it was David, though. He won the Nobel Prize for this. Significant amount of work. So, of course, he classified them, right? 1 through 7. Of course, it's the lovely Roman numerals to boot. But the good news is I'm not going to make you memorize this, right? You can look this up, right? It's just knowing that this exists, okay? So this table I like, though, because it gives us all the examples, all right? So first, we have double-stranded DNA is one of the possibilities, right? And the human herpes viruses fall into that category. Single-stranded DNA are these parvoviruses. Double-stranded, and notice we went to RNA, and then there's a DNA down here, because this is double-stranded DNA genome with an RNA intermediate. And this happens with the hepatitis B virus. So there's some interesting things that happen here. So here's the single-stranded genome, and notice they write that it's positive sense, right? So this single strand of RNA can be directly read by the ribosome. So this is polioviruses are like that. And of course the double-stranded RNA viruses. Single-stranded, but look, negative strand. The influenza virus is like that. So it, has this problem, right, that its RNA is the wrong direction for our ribosomes to read. And then good old HIV, which is why I'm going to play, because we love this one. But this one I think is really interesting. So it's a single-stranded RNA genome. They don't tell you whether it's plus or minus. It doesn't really matter because guess what? It forms a DNA intermediate in its replication process. So I found us a video in our book even that I think is worth watching. I think it's worth the 3 minutes. So go back to the book, Erica. Go back to chapter 8.3. That's why I made the note. Section 3. How do they replicate, right? So this is a diagram we'll look at in a moment, right? That shows you the replication strategies for that classification. But I want HIV, that's pox. It's probably towards the bottom of the page, right? Negative sense. Here is HIV. So there is actually an animation that goes along with this one. And it starts out with a horrible picture. Gotta turn it up.
But HIV is even smaller. The complete virus.
Sorry about
the
that.
complete virus. AIDS is caused by HIV. HIV's host cell is microscopic. But HIV is even smaller. The complete virus, or virion, consists of an envelope, capsid, 2 strands of RNA, and enzymes needed for replication. The envelope has numerous GP120 glycoproteins embedded in it. HIV causes AIDS by infecting and destroying important immune cells that possess a CD4 receptor. The GP120 of the envelope adsorbs or binds to the CD4 receptor and co-receptor on the host cell, like a key.
So that's membrane fusion, right? For an envelope virus. Did you see that? That's how that entry happened?
He fits into a lock, and the virus enters the cell. Reverse transcriptase reads the RNA, making a DNA copy of it.
So that enzyme it comes with, reverse transcriptase. And actually, viruses like this have led to really great genetic research because we stole this virus, right? But we don't normally go from RNA to DNA, right? Our process is DNA to RNA to proteins. But because they are an RNA, they come with an enzyme that can turn RNA into DNA, right? That's why it's called a retrovirus, right? Because it's reverse transcriptase, right? We're going in the reverse direction. It's an important point. Does that make sense to y'all? Okay. I'm going to keep it going. Making that DNA.
AZT and other reverse transcriptase inhibitors slow this phase of HIV replication. Using the viral enzyme integrase, the DNA joins or integrates into the host cell chromosome, becoming a provirus. Integrase inhibitors are targeted against this phase of replication. The provirus is latent for an indefinite period, then starts making new viruses. Messenger RNA is made for the envelope proteins, The proteins are made by ribosomes and move to the cell surface.
Really? Do we have to do that more than once? Okay, apparently. Sorry.
Messenger RNA.
And he didn't even tell you what he was doing.
Is transcribed.
All right. That's okay. You can tell I didn't watch this ahead of time, right? Sorry, guys. Okay, so look. These are those viral proteins that are important for attachment. And notice it makes it and then it sticks it into our cell's membrane. Okay? It's an important step he didn't explain. All right. Just label it.
Messenger RNA for other proteins is transcribed, then translated into a large protein.
How many times are you going to do this? So what do you think they're making now? So those were the ones that go into the envelope.
Viral protease cuts this protein into several pieces, forming the capsid, protease, reverse transcriptase, and integrase.
That's interesting, right? So they make this big, long messenger RNA, right? They make this big, long string of amino acids, right? But it actually makes 4 different proteins, right? So they come with an enzyme that cuts them into 4 different proteins. One of them is the capsid, right? That's going to surround the nucleic acid when it makes it. The protease is so it can keep doing this, right, when it infects cells. And then the reverse transcriptase, right, that turns their RNA into the DNA so it can integrate into our cells, right, hijack our system at that point, right? And, of course, they need proteins in order to do that, right? Those integral proteins they mentioned is how they take their DNA that they made and stick it into our DNA. It's pretty crazy, huh? All right, so now keep going.
Protease inhibitors target this process of cutting the protein into functional parts. RNA copies of the provirus are now made and combine with the proteins to form viruses.
So now it's making its RNA for the virus. And then self-assembly of the nucleocapsid.
Virions pick up their envelopes during release from the cell.
So that's pretty cool. So you notice a couple of times in this video he said inhibitors, right? So we know we have drugs nowadays to help stop the parts of this cycle. So he was mentioned when he said protease inhibitors, right? So some of those drugs that people who have HIV are taking interfere with that protease. So it can't make those proteins, right? And so he mentioned a couple others. I'm not real versed on HIV treatment, but there are several drugs now that interfere with different, obviously it makes sense that how it works is it interferes with different steps in this process of making the HIV virus. So this is a lot, huh? I'm not going to ask you specific questions about this process, right? This is not a virology class. But it's important to realize the steps that some of these viruses go through can be quite complex because of the type of nucleic acid they have and how they achieve the process of copying themselves, right? That synthesis inside the cell. And of course, you know, HIV is a good one to pick because it's so personal, right? Everybody knows about HIV and how horrible it is, right, to one's immune system. Okay, so not freaking out, right? Okay, I think that was a really good animation. Other than him not saying what these were going and just being labeled, that kind of irritated me. But other than that, he gets an A on presentation. And so there are other possibilities for other viruses, right? So I don't think they did, so retroviruses, right? So HIV, as I said, is referred to as a retrovirus. And unfortunately, you know, some terminology becomes used so much that people forget what it actually means, right? But this has to do with that reverse transcriptase test step. And so this is a great diagram too that shows the different steps too. It's a really good one. All right, so negative strand. Let's see if I can pull this figure up bigger. Yeah, okay. So here is negative strand, right? And they're not even showing you how this one gets in. But we know, right? This is a naked virus, right? I don't see any envelope on it. So it got in by how? Endocytosis. And then it uncoded, right? And so notice that it comes with, right, viral RNA dependent RNA polymerase. So because it's minus strand RNA, it comes with its own enzyme to make the plus strand. Because again, we don't do that in our cells, right? We don't even make negative RNA, right? We make positive messenger RNA. So they've got an extra step they've got to do, these types of viruses. They've got to turn, right, they got to come with their own enzyme that turns their negative RNA into plus RNA. So that's now the same as messenger RNA. So our ribosomes can attach to it and make their proteins for their protein coat. And then some of it, right, it just gets copied by their enzyme into more of their negative strand. And then assembly happens. Make sense? So this you do need to know, right? That if you're a negative strand RNA, the additional step and the additional enzyme they have to come with themselves is an enzyme that turns negative RNA into plus RNA, which is the same thing as messenger RNA. And then they can hijack our system, right? Use our ribosomes to make their proteins. And of course, they're using our nucleic acids to make their make their RNA, right? Questions? Okay. So those are we started with the hardest ones, right? I'm going up to the easier ones. Up the chain to the easier ones. So this must be our plus strand one. And they actually have an animation for the plus strand one. But we're just going to look at the figure. Where's my link? There's no link? There it is. All right, so, okay. So, right, endocytosis, they're even showing you here, naked virus. But this one is plus strand, right? But this is one of those weird ones, right? Where look, it comes with, again, an RNA-dependent polymerase. And for some unknown wacky reason, they make negative strand RNA. And plus strand. They go back and forth. Even though they're a plus strand, right? They make a negative strand intermediate.
You said it depends on what direction they go in, depends on guarantees the negative and positive.
Yeah, the direction of the RNA. For RNA and DNA, directionality matters on how enzymes can read it. So ribosomes can only read plus RNA. It's the same thing as messenger RNA. So look, this virus, right, is plus. So our ribosomes would be able to attach to it right away, make its viral proteins, right? It just needs this enzyme to make more copies of itself. But that's in lies the problem, right? Because think about this. With DNA, we've got 2 copies, right? It's double-stranded for us. So the reason why they're doing this is even though this can be read to make the proteins, right? So notice that from here to here, you could go right straight to protein, right? But why is it having to make, right, a negative strand of RNA? Why is it having to do that? So think about our cells again. Do we copy RNA? No, we make RNA from what? DNA. There's no DNA here, right? All it is RNA. So in order to make copies of their own RNA, they've got to make the minus strand to make copies of the plus strand from it. Does it make sense now? Why they do that step? Why they have to do that step? And they got to come with that enzyme because we don't do that, right? We don't copy RNA. We don't make RNA. Did I give you an example of what type of virus does this? No. It's probably on our list, right? And of course, I can't even tell you what number it is on that chart either. They don't even repeat it. Okay, so moving up, let's now look at this one. Okay, so this is easy peasy, right? So what's happening here, right? How's it getting in? It's endocytosis, right? And then it's probably going to uncoat, and that protein and the DNA come out. And this is another one that comes with its own RNA polymerase. Okay, so here it is, double-stranded DNA, right? But it's going to make its own, it has its own RNA polymerase. So from its DNA, it makes messenger RNA. And that can be translated by the ribosomes, right, and made into their protein coat. And then they could just use our enzymes, right, to copy their DNA into more DNA. So that's the easiest one, right? Why do you think they have to come with their own RNA polymerase, though? Look at where this is happening. In the cytoplasm, right? Where do we make RNA in our cells? In the nucleus. Because this DNA, unlike HIV, doesn't go and incorporate into the nucleus. It doesn't have access to our RNA polymerases. It has to come with its own. But apparently, DNA polymerase, I don't know, outside the cell, outside the nucleus, DNA can be copied. No. Yeah, RNA polymerase. Okay. Pox virus. Example of that. Okay. So now, what's this one? Oh, this is just general replication. Where it goes into the nucleus. Okay, so DNA viruses, straight up DNA viruses. Okay, so here's the easiest one, right? So look, this is an envelope virus, right? What's happening here? How's it entering? No, it's membrane fusion. It's envelope, you see, it's fusing with our membrane. Okay, then it uncoats, and then look, where does that DNA go? Into the nucleus, right? So it can use our RNA polymerase, right, to make its messenger RNA, to make its proteins. And then, again, make copies of the viral DNA. And the interesting thing here, look at with this one. The proteins come back into the nucleus and it assembles inside the nucleus. And then they have to leave the nucleus and then leave the cell. Kind of cool. So that's the simplest way for them. I think if I hit that X, it's going to be the wrong X. All right, so then you get to this chart, which is in my PowerPoint. Right here. This is just the DNA ones. Okay? So that one is this is the one that we just looked at, right? So if it's a DNA virus, class 1, right? It goes into the nucleus. We'll copy its DNA for it. We'll make its messenger RNA for it, right? The other thing, right, for DNA viruses is sometimes they'll come in single-stranded. That's fine, right? We can take a single strand of DNA, make double-stranded DNA, right? And digest away one of those for making the virus. And we can use the double strand to make messenger RNA. We didn't go through that example in any of the pictures. What virus does that? So what is this one? Class 2? Parvoviruses. It's an example of that. Okay, so this one, double-stranded DNA, right? So right from the DNA, we can make messenger RNA, make those viral proteins, right? But this one, right, goes from DNA to RNA back to DNA to copy its DNA. So it has that reverse transcriptase. That one is, did we look at that one? No. That one is hepatitis B virus. They didn't have an example of that one. But that one's kind of funky. You're like, well, why would you do that? You don't really need to do that, right? Your DNA, why are you going RNA to DNA? And my belief is, right, and it probably has something to do with when we look at the RNA virus, especially when we look at HIV, right? Okay, so RNA classes, 4 of them, right? If it's double-stranded, no problem, right? You can digest away, have that plus strand, make those proteins, right? But you've got to make your, they make a single strand to make their double strand. What ones are those? I should write these on the slide. So this one is 3. Who does that? Reoviruses. I don't even know any of those. All right. So, influenza, though. We looked at that one. Did we look at that one? I'm losing track of what we looked at and what we didn't look at. But you can see, right, why he ended up with all these different classifications, right? This is the easiest one. This is where it gets more complicated, right? When you're minus strand and you have to make your own plus strand and then double-stranded back to single-stranded for replicating your RNA. This one, right, single-stranded plus stranded RNA, right, but it makes double-stranded DNA, right? Which then they make messenger RNA from, and they can make more of their RNA. But what happens here with this double-stranded DNA for this class of virus, this class, I hate Roman numeral 6. This is HIV, y'all. Yeah, it integrates into the host genome. And this is why I wanted to go over this before I go through a similar process that can happen in bacteria. And this is called lysogeny. So HIV does this, but other viruses do this too, where they literally, right, integrate their DNA into our chromosome. So they virtually hide in your cells. So this is the major problem too with HIV, right? And in other viruses like your herpes viruses, and one of them most of us are aware of, and maybe even had as children. If you're old like me, most children don't get chickenpox anymore. Anyone know why? In 1995, they came out with the vaccine, right? But guess what the vaccine is? It's the virus. They give it to you. It incorporates into your cells, right? You just don't get the infection. And then later on, it can pop off the chromosome and replicate again. And that reactivation we call shingles, right? So I'm out of time this time, but which is good, right? But I'm going to post an animation, this one, for bacteria, for that process, right, of integrating into the chromosome, right, and popping off, for y'all to watch for homework, okay? I'll put it right as an announcement.